Hi, my name is Kiana, and I'm the co-founder and CSO of Kadia Bio, and I'm also an assistant professor of biomedical sciences at Keck Graduate Institute, a member of Claremont Colleges. In this presentation, I'm going to introduce to you the vision of our startup, Kadia Bio, our platform, and our first line of products, which are designed for CRISPR quality control applications. So, just make the slide. So biology is systems of systems, which are continuously communicating with each other in a dynamic fashion. But for many years in life sciences, we have been taking snapshots of these dynamic events using optics to understand biology and to identify different pathologies. To truly understand biology, we need to understand the interplay between these systems in a dynamic way. And biology is comprised of lots of these dynamic systems that are connected. For example, nucleotides, protein complexes, small molecules, enzymes, cells, and various tissue types. So biology is systems of systems. So to truly understand biology, you need to take a system biology approach. We need to have an insight to multiple analytes and multi-perspective biological data in near real time, where we can have a flow of information, not just a snapshot data sets. Having an insight to multi-analyte activity in real time is the only way to understand biology and the interplay between these different biological systems, which can pave the way for new fundamental discoveries in biology. So this vision requires obtaining large volume of data, including genomics, proteomics, and other type of biological data, and sometimes in a personalized fashion. Obtaining this real-time multi-omics data using existing technology like optical platform is not possible. The wavelength of optical platforms are limited, and it's not sufficient to look at nanoscale biological events. Quantitative multiplexing will require expensive instrumentation if using optics, amplification, and many optical labels, which makes the process very, very complex. So we need to really have an infrastructure where we can measure multiple different analytes at the same time and be able to transfer this large volume of multi-omics data to computers, so we can develop algorithms to process them and to unlock their immense amount of information. This is only possible through highly scalable, cost-effective electronic system that can also be personalized and seamlessly transfer data to computers for analysis. And this is what Kadia does. Kadia is connecting biology to computers for the very first time by building an infrastructure using transistors to directly stream multi-omics data to computers in a near real-time fashion. This allows a new generation of platform to obtain new biological information. Our platform works by utilizing graphene-based field effect transistors. In these transistors, a conductive graphene channel is produced between the two terminals of the transistors, namely source and drain. And the third terminal, which is the gate terminal, controls the electric field applied to our transistors. The graphene channel is biologically activated to specifically interact with our target analyte, which can be a protein, a DNA sequence, or any biological molecule. The interaction between the target analyte and the biology fixated atop our graphene channel changes the conductivity of the graphene material, which in turn changes the electrical parameter of the transistor, which can be monitored in real time. Also, graphene-based transistors are superior to silicon-based transistors, as graphene is biocompatible and can directly be connected to biology without impacting it. Also, biology doesn't impact graphene. In addition, due to the graphene high electron mobility, this transistor can transduce the signal from biological event much faster. So our core technology enables us to be rapid in detection. It also provides us with quantitative analysis and not just qualitative results. The sensing part of our platform can be reconfigured in terms of the analyte that we can detect, ranging from DNA, RNA, enzyme, antibody, or small molecules, and in terms of form factor that is best suited for each specific applications, ranging from handheld system to an automated robotic systems. And all the data can be transferred seamlessly to computers for analysis. We have been previously developed and published a variety of analyte detection platforms using our transistors in collaboration with our academic and industrial partner. This includes the detection of Zika virus in collaboration with CDC investigators, aging biomarkers based on antibody-antigen interaction in collaboration with the University of California, Berkeley scientists, small molecule detection and cytokine detection in collaboration with our industrial partner. Our collaboration has enabled us to further improve our technology for each specific application, and we will continue to do so. It also enables us to work with our collaborators and partners and combine our expertise to develop better products. So last year we published a technology that was using CRISPR as a detection platform. Our transistor utilized the search function of the CRISPR, and we're able to seamlessly detect when CRISPR molecule finds and binds to its target DNA without the need for amplification or basically amplifying the target. This was the first time that an interaction of CRISPR complex with target DNA sequence was captured with an electronic transistor. The technology called CRISPR Chip was published last year on the cover of Nature BME and combined basically three revolutionary sciences, transistors, CRISPR, and graphene. So after publications of the CRISPR chip, we have attracted many people that were using CRISPR for a variety of applications, and we are actively working on them, we have already partnered with them or established a partnership with them to develop better tools to monitor the efficacy of the design of CRISPR-based therapeutics. Basically for various different applications, for therapeutics application, for gene modulation, imaging, and many applications. And our technology was basically was used as a quality control, which I will talk about more in the next few slides, as well as establishing partnership to use our platform for amplification-free disease detection, as well as monitoring genetic modifications in plants. So briefly, Cas enzyme in our CRISPR chip technology, Cas enzymes are mobilized on the surface of the graphene. Guide RNAs are then introduced to program the Cas enzyme to detect the specific DNA sequences, and then the DNA sample is introduced. The system can monitor the interaction and binding of the guide RNA to Cas enzymes to form a stable CRISPR complex. In addition, it can monitor the interaction of the CRISPR complex to target DNA without the need for amplification. We have used our CRISPR chip technology for a variety of different applications: detection of deletion or insertion without amplification in human genome. We have done this in some genetic diseases, including Duchenne muscular dystrophy disorder, as well as detection of single point mutation without amplification in sickle cell disease. We were able to use the technology for comparing the efficacy of guide RNAs with different lengths in binding to target sequences. We were able to compare the efficiency of various different Cas versions in binding and targeting sequences. And also detection of RNA binding to Cas proteins in high throughput automated system with software package for data analysis. So what you see here is the result from our previously published work, which indicated the ability of CRISPR chip in detection of 2 exon deletion commonly seen in patients with Duchenne muscular dystrophy disorder. 2 different chips were designed. One was designed to target exon 3, and one was designed to target exon 51 in human dystrophin gene. The chips were able to detect the presence or absence of each of these exons in human genomic samples without the need for amplification. We have done the same thing with sickle cell disease, where we were able to detect the mutation in HBV gene in human genome as well without amplification. The power of real-time data is really important. For quality assurance purposes, the whole process of chip functionalization is monitored in real time. This was really important as we found out about this after we developed the technology. During the functionalization process, we first immobilized the Cas enzyme, and then we introduced the guide RNA to program the Cas for a specific targeting. We actually monitor in real time to ensure that the guide RNA is bound to Cas enzyme as a part of our quality assurance. It turned out that monitoring the guide RNA binding to the enzyme is very important as enzyme modification, Cas modification, environmental factors such as pH, temperature, buffer composition, and all of this could impact the binding of guide RNA to Cas and the stability of the complex. We were able to monitor all these events in real time from our technology, and from this our first product was actually born. So our first product, which was developed in collaboration with Kobo Technologies, pioneering in CRISPR QC, is a product to monitor the binding of Cas to guide RNAs. The product utilizes an array of CRISPR chips within a liquid handling system for CRISPR quality control applications. The first product, CRISPR-binds, monitor the interaction between Cas enzyme and guide RNA to ensure successful complex formation and stability in different environments. Basically, the system can be used to monitor the interaction of a variety of engineered Cas enzyme with synthetic or chemically modified guide RNAs, which is a critical step in designing effective CRISPR complexes. So the video that you're going to see here shows how CRISPR-bind works. This is, of course, our minimum viable products, and the future version will be built with features and modules that our partner require for their specific application. The user can select a protocol, enter information about guide RNA, Cas buffers, and other testing conditions that they would like to see, such as pH, temperature, and evaluate the binding and stability of the complex at these various different conditions. The system is fully automated system, which can eliminate user variability while providing a high throughput format. This can also provide a tool to see the impact of different parameters on complex stability using machine learning and learn more about the biology of CRISPR itself. Okay, I apologize for the silence. I was not able to talk over the video, but what you saw in the video, basically the data at the end of the assay that was performed by the system, the data was transferred to a computer for analysis, and the data will be then analyzed in a few minutes, making the process rapid and accurate without any user variability. Using CRISPR-bind product, CRISPR design can be optimized to achieve the desired outcome. The plot that you can see here shows two guide RNAs with two different binding efficiency, which were measured and analyzed with our CRISPR-bind product. So the next generation of CRISPR quality control product built on our CRISPR Chip technology includes CRISPR-Amp, which monitors the efficiency of the designed CRISPR complexes to amplicons to select the best CRISPR complex. This product will allow you to select the best combination of Cas and guide RNA for maximum binding efficiency, which is very important in applications such as CRISPR-based gene modulation and imaging. Similar to gel electrophoresis, but much easier, much faster, with quantitative result in a high throughput format. Future product will enable quantitative monitoring of cleavage as well, providing us with the full picture of CRISPR activity on target sequences. Another pipeline product is CRISPR Gen that monitors the interaction of CRISPR complex with the whole genomic sample, eliminating the need for amplicon generation and amplification, making the whole process significantly faster. The plots on the bottom right indicates that CRISPR Gen can distinguish the binding efficiency of 2 guide RNAs with target sequences with single point mutation. The result, the purple plots, show that the guide RNA with 24 nucleotides in length binds less effectively to the target sequences that have 22 nucleotides. The results were analyzed with machine learning, and as you can see here, the result is comparable with the gel electrophoresis result, but without the need for amplification. And by simply just using the whole genetic sample, which significantly reduces the time that we require to generate those amplicons and also the time for analysis. The next generation will also be able to monitor cleavage activity. The product can be also utilized to monitor the efficiency of gene editing before and after CRISPR editing, which can significantly facilitate monitoring CRISPR-based editing efficiencies. And we are currently partnering with various different industries to bring other products utilizing our technology. With that, I want to thank you for listening, and please contact us if you have any questions or if you're interested in partnership. Thank you.
Okay, great job.
Yeah, so what's going to happen, because I think I didn't know how to, I forgot I have to go back to these slides.
Mm-hmm. Yeah, I know that there was a little bit of a pause after the video, but I made a note so I can edit out the pause and just start it again when you pick back up.
Perfect. Okay, thank you so much.
Of course. Any other questions for me before we wrap up?
No, and thank you really for accommodating this. I feel much better about this one than the one yesterday. Of course,
yeah, no problem at all. And once we have the editing done, I can send over another preview link if you want to take a look.
Perfect. Thank you so much.
Thank you. Have a good rest of your day.
You too. Bye-bye.